everyone. Thanks for joining our webinar. My name is Kasia Proctor, and I'm the Director of Marketing Communications here at Biolog. I will be your moderator today. Our webinar today is titled Harvesting Insights for Sustainable Agriculture, New RhizoPlates for Advancing Microbial Analysis of Nitrogen Fixation and Phosphate Solubilization in the Rhizosphere. First, I have a couple of housekeeping items. This webinar is being recorded, and we will be sending the link to anyone who has registered. That should be coming out in probably a day or two. If you have questions throughout the webinar, please submit them. At the bottom, you should have a Q&A chat feature. Many of you submitted questions during the registration. Thank you for that. We will try to get to those either during the presentation or at the end. And then if you come up with more questions, then again, the Q&A, please. Okay, now let me introduce our speaker. Juan Sanchez is the Consumables Product Manager here at Biolog. As a former Stanford scientist, he co-authored several microbiome publications featured in Nature. He also has previous experience in sustainable agriculture. Juan, please take it away.
Thank you, Kasia. Good morning, all. Today we have an exciting topic, Biolog's new RhizoPlates, targeted at sustainable agriculture for measuring microbial nitrogen fixation and phosphate solubilization. So why are nitrogen fixation and phosphate solubilization important? Well, they're very necessary for crop growth in order to produce food. In fact, companies are investing billions of dollars to supplement these as fertilizers. However, this has had detrimental effects on the water, the soil, and it causes some greenhouse gases as well. To add to that, it's not the most cost-effective solution, with very little of the phosphorus from mining going to the plants. Fortunately, microbes hold the key with their ability to fix nitrogen from the atmosphere and solubilize inorganic phosphorus to solve this issue. The real challenge now is finding the best microbes, and that's where Biolog's technology shines. Our company has been involved in this area for years, particularly with the EcoPlate for soil, water, and waste analysis, and it has contributed to over 500 publications. We surveyed our extensive customer base and learned that nitrogen fixation analysis and phosphate solubilization analyses were critical underserved areas where very laborious, outdated, and difficult technology was in place. RhizoPlates address all of these issues, enabling the assessment of nitrogen fixation and phosphate solubilization performance with under 20 minutes of hands-on time, a true game changer for the sustainable agriculture industry. Here's a screenshot of Odin doing nitrogen fixation performance for multiple strains. High curves indicate high performance. Nothing is easier. The differences are clear. Now let's talk about what people are currently using to analyze N fixation and P solubilization. First is the acetylene reduction assay. This assay uses nitrogenase activity through the reduction of acetylene gas to ethylene, and this is analyzed by a gas chromatography. It's worth noting that acetylene reduction assays do not directly quantify nitrogen fixation, but use acetylene as a proxy. Advantages for this assay are its high sensitivity and that it provides a quantitative comparison. However, there are some disadvantages here, particularly that it is very labor-intensive and time-consuming. It also has a limited dynamic range where high concentrations of acetylene can be toxic to microbes, and low concentrations can produce undetectable ethylene production. And lastly, it uses acetylene gas in sealed containers, followed by gas chromatography. So these two processes are very cumbersome. I don't imagine the scientist in this picture is going to have a very good time today. Now for phosphate solubilization. Here we have PVK plates. These plates contain insoluble inorganic phosphate, and Phosphate solubilizing microbes can release organic acids or employ enzymes in order to help solubilize this phosphate and make it biologically available. When they do this, they generate these clear halos around the colonies. And in order to measure the assay, scientists look at the size of the halo to see how much of an ability to solubilize the microbe had. Some advantages here is that it is a very low-tech technique. It can be deployed in any area, and it's capable of screening large number of isolates. However, there are some disadvantages, such as it being only a qualitative technique, and it has limited compatibility. Not all microbes will be able to grow on this PBK plate. Additionally, expert interpretation is required because there are several factors that impact the size of the halo, so not the perfect solution. Let's veer away from the status quo to phenomics, technology we adapted to measure nitrogen fixation and phosphate solubilization. How does this work? Well, cells consume food. The food goes into several pathways, which result in cell growth. If we control what food we make available and substitute the nitrogen and phosphorus with diatomic nitrogen from the atmosphere, which is usually inaccessible, and inorganic phosphate, which is insoluble, then we can control the food and constrain how the cells will grow over time. This will help us discern which strains are better able to use the nitrogen or insoluble fixation based on growth. We combine this technology with Odin, a high throughput solution for dynamic incubation and measurement of up to 50 plates. It is also offered in a smaller 8-plate version to accommodate the user's needs. It's supported by its own intelligent software, which computes multiple parameters for kinetic characterization and comparison, resulting in better results and more actionable data. Our platform leverages the power of Odin integrated with RhizoPlates to offer a comprehensive and advanced solution for detecting, ranking, and monitoring nitrogen fixing and phosphate solubilizing bacteria. Incubation and kinetic data collection is supported on Odin. Additionally, our platform supports community analysis, a critical component needed to understand the interactions such as cross-feeding behavior between constituents of the soil microbiome. All of this can be done at high throughput, delivering easy to interpret, streamlined reports for seamless analyses. Now onto the RhizoPlates. But we can't talk RhizoPlates without talking about the rhizosphere. The rhizosphere is a narrow region of soil surrounding plant roots. It's populated with microbes and regulates many important processes, including nutrition from nitrogen and phosphorus, among other things. We have two RhizoPlates, one for nitrogen fixation and one for phosphate solubilization. First, let's look at RhizoPlate N. It contains 30 carbon substrates relevant to nitrogen fixing bacteria. In this plate map, you have your organic acids, which would be plant-derived in soil environments. Lots of citations for those, as well as simple sugars like fructose common in root environments. You won't find any amino acids here. All substrates are devoid of nitrogen, forcing that nutrient to be derived from the atmosphere through nitrogen fixation. Both of the plate maps for RhizoPlates can be found at biolog.com. Some features of this plate are its 30 different substrates pre-plated in triplicate to simplify stats. This lets us see which carbon sources, isolates, or communities use better. and we're able to find the best carbon source, as well as holistically account for the performance of all substrates through a new parameter I'll talk about later. The plate is simple to use. Just inoculate your cells in our nitrogen-free inoculating fluid and start. Automated analysis can span 7 days, enabling measurement for even slow-growing soil bacteria. And kinetic growth data will give a larger dynamic range than acetylene reduction assay. Lastly, the streamlined protocol reduces manual labor 10x compared to acetylene reduction assays. In a similar 30 substrate format is the plate map for RhizoPlate P. Here, you will definitely find those relevant amino acids we left out from the other RhizoPlate, not only important for biomass formation, but also documented to engage in metabolic processes for P-solubilization. Some of the same organic acids available in the roots are present here, but notably sugar phosphates have been left out as they contain phosphorus. None of the RhizoPlate P substrates have any phosphorus, forcing that nutrient to be derived from insoluble tricalcium phosphate pre-coated uniformly throughout the plate. Let's look at the features. 30 different substrates are featured here again in triplicate for easy stats and individual or aggregated assessment of soil, isolate, or community performance. The plate is simpler to use than even PVK, with the insoluble phosphate conveniently pre-coated. Just inoculate the phosphorus-free with the phosphorus-free inoculating fluid and start. The assays can be conducted for 7 days, and the kinetic growth data enables relative quantitation, unlike subjective PVK plates limited to qualitative analyses. Let's explore some use cases. First, screening isolates. RhizoPlates enable us to look at a massive collection of bacterial isolates, or just a handful, screening to find the best performer nitrogen fixer or phosphate solubilizer, and allowing more rigorous in-plant testing with the best candidates. RhizoPlates also work with communities. Complementary species can be combined for broader substrate utilization, or complex soil samples can be inoculated and analyzed on the plate, all of which can be done with Odin on a single integrated platform. And finally, for those of us growing our food, thank you, RhizoPlates can be used to monitor the persistence of nitrogen-fixing bacteria as well as phosphate-solubilizing bacteria in the agricultural field over time, ensuring optimal dosing and making the application of biostimulants and fertilizers more cost-effective. Here's what Cultivarium, a company developing novel tools for engineering non-modal microbes, had to say after using our product with their soil isolates. The RhizoPlates complemented Cultivarium's predicted computational pipeline, assisting with the ranking of nitrogen-fixing and phosphate-solubilizing bacteria, helping to guide their engineering efforts. We're also proud to share feedback from Earnest Agriculture, a company providing seed coatings containing microbial consortia to enhance crop performance. Earnest Agriculture was already using Biologs Gen 3 and PM plates on Odin, and are now leveraging RhizoPlates as well to easily and rapidly evaluate microbial isolates and communities. How do RhizoPlates compare with the status quo? For this assessment, we enlisted the help of one of the world's largest agricultural companies, which had extensively characterized strains, including data from PBK, ARA, sequencing, and beyond. Let's look at some results tabulated using Odin software. We can see clear phosphate solubilization rankings for all 10 of the strains tested. These were achieved using relative quantitation values from a new parameter, average well growth development area under the curve, which we will discuss more later. With this, not only do we match PBK ranking, but we also get better resolution. Differentiation of the top 2 strains was achieved, which was not clear using PBK. Mid-performers were also unraveled. And for the bottom 3, we were able to find phosphate solubilization in strain B, which was supported by sequencing data, but could not be demonstrated on the PBK plate. For nitrogen fixation, we can also see clear rankings and excellent degree of separation between the top performing strains. Other than an inversion for strains F and E, which yielded very similar AWGD AUC results, we matched the ARA rankings. For F and E without directly measuring nitrogen through a cost-prohibitive and impractical isotope-based analysis, it is impossible to discern which of the 2 rankings is correct. We also observed RhizoPlates' higher dynamic range, as we're able to detect nitrogen fixation in strain H, which was below the limit of quantitation for the acetylene reduction assay. Beyond being impressed with RhizoPlates' performance, our collaborator remarked on the great time saving for this assay, taking minutes rather than tens of hours of wet lab work. Here is a quick overview of the RhizoPlate protocol. Samples are inoculated on either RhizoPlate with their companion inoculating fluid. They are loaded on Odin, where kinetic growth measurements are taken. And finally, average well growth development plots are produced to rank strain performance. Very simple. Here is an isolate focus protocol for RhizoPlates, which can also be performed with communities as well as soil samples. Note how short it is, only 5 steps, no complicated steps dispensing insoluble phosphates or working with sealed containers with acetylene gas and then having to sample these for GC. The main takeaway here is simplicity. Just inoculate the RhizoPlates, load on Odin, and walk away. Once you walk back, you will notice Odin also performed QC and treatment of the data, making analytics, ranking, and candidate selection a breeze. Now that we've covered the basics, let's look at a case study analyzing both nitrogen fixation and phosphate solubilization in soil isolates provided by Cultivarium. Odin was used with both RhizoPlates, looking at 8 strains per experiment. The only hands-on wet lab timer was during setup at the beginning of the experiment. Here are the 8 strains encompassing positive and negative controls for both nitrogen fixation and phosphate solubilization. Additionally, there are 4 biostimulant candidates included, which had predictions for nitrogen fixation and phosphate solubilization. C1 through 4. Here is a screenshot of the data from Odin comparing two positive control strains, P. putida and A. piti. Odin performs segregation and background subtraction for you with the click of a button. We see many wells with clear growth, indicating they were able to use the only phosphorus source, insoluble tricalcium phosphate, and the single carbon substrate in each well. Here we can see significantly greater growth with lactic acid by A. piti than by P. putida, which is not matched by other organic acids in this assay, denoting each organism's unique needs and enabling synergistic strategies for building synthetic communities capable of leveraging everything in the soil. We can also see growth across the plate for RhizoPlate N for both C3, which had predicted nitrogen fixing abilities, and A. brasilense, our positive control, indicating they are able to use diatomic nitrogen as a nitrogen source, thus nitrogen fixing. Here we also highlight lactic acid, which both RhizoPlates share, with A. brasilense growing significantly better with it. Next, we can see the variation among replicates for different substrates of interest for C3 in the context of nitrogen fixation. Candidate C3 was predicted to have both nitrogen fixation and phosphate solubilization activity. When we look at C3 in the context of phosphate solubilization, we find an entirely different set of substrates implicated, indicating the diversity in the mechanisms and pathways used for nitrogen fixation and phosphate solubilization by the same organism. And by looking at C1, a different strain, with predicted nitrogen fixation, we uncover a different set of implicated substrates, uncovering more potential synergies for building synthetic communities to enhance coverage amongst the substrates. From these visualizations, however, it's difficult to rank nitrogen fixation or phosphate solubilization performance, accounting for all 30 substrates. To try and gain a clearer picture, we leveraged the popular tool for reducing the dimensionality of data. In this principal component analysis, using the kinetics data for all 30 substrates as loadings, the positive and negative controls are somewhat separated across the first principal component, which accounts for 46.3% of the variation. However, it's difficult to infer anything further, such as rank. A better way of performing data reduction for actionable insights is available in Odin via AWGD analyses. Let's look at phosphate solubilization simultaneously for our 8 strains. Here is a screenshot of average well growth development from Odin, AWGD. Each point plotted accounts for all the different carbon sources, aggregating them into a single read to evaluate strain performance. Even with the naked eye, it's easy to see which strains perform better, with A. piti coming out on top. P. putida in teal and C3 in brown underscore the importance of Odin's kinetic rates, as they reach very similar AWGD levels at the endpoint, despite a higher curve for P. putida prior to 75 hours. Omitting such detail could lead to the wrong ranking. Even though this is easy to see by eye, Odin automates this further by calculating and tabulating the AWGD area under the curve for all strains. Matching the AWGD plot, we have A. piti with an AWGD value of 17.1. This is clearly separated from P. putida in second place at 12.16. And even for P. putida and C3, which intercept at 75 hours and share similar endpoints, we can make a clear determination on how they rank. These nitrogen-fixing strains are clearly separated from the negative controls like S. epidermis and E. coli. For nitrogen fixation, we can see similar differences with greatly stratified endpoint measurements. It's clear to see we had top performers, which are greatly separated from our negative controls. Ranking by eye here, particularly among the top two strains, C1 and A. brasilense, can be a bit challenging, which is again another area where Odin helps catching the subtle differences. The top two strains are distinguished here based on their AWGD area under the curve values, which are tabulated in Odin. Although C1 showed the highest AWGD at the endpoint, A. brasilense slightly outperformed it. Additionally, with our negative controls showing near zero values, we observed that the two other strains also demonstrated significant nitrogen fixation. However, this is only part of the story. With the full data in hand, we can now see that one strain predicted to exhibit both nitrogen fixation and phosphate solubilization performed exceptionally well in both assays. This strain would be a strong candidate for a biostimulant product. Now, before we announce the winner, let's review the key takeaways. Our customers asked for better tools to save the earth, and Biolog delivered with its game-changing RhizoPlates for smarter, sustainable agriculture. We demonstrated major differences in substrate utilization, revealing sample stratification on our AWGD plots for next-level insight. We showed that kinetic monitoring is key in correctly ranking strains. We rank strains based on the AWGD area under the curve values, identifying the true champion of the test. Candidate strain C3 emerged as our winning strain, with high levels of both nitrogen fixation and phosphate solubilization. Lastly, we made the case for exploring communities of strains with diverse substrate preferences as the key to unlocking the next wave of biostimulants. Beyond the products we discussed today, Biolog also has solutions for the identification of microbes, including gram-negative, gram-positive yeast, anaerobes, and filamentous fungi. We also carry chemical sensitivity assays and support mammalian cells. For ecological needs, our EcoPlates are compatible with soil and water sample analyses. And if you have a unique use case, we have custom microplates to offer a solution. Now, recently we acquired anaerobe systems, merging our expertise in working with anaerobic microbes. Please check out our website if you're interested in learning more about our anaerobic media and chambers. The Odin line of instruments and intelligent software support all of these products, simplifying research at any scale. And lastly, should the need arise, our US-based ISO certified lab is capable of offering Odin-based phenotypic services, including RhizoPlate analysis, as well as ID solutions like sequencing and MALDI-TOF. Thank you for tuning in. We hope you found this insightful and look forward to hearing how this new solution powers your analysis pipeline.